Hello students, in last video and in last classes we have completed our chapter number 5, the principles of inheritance and variation, in which we discussed about the different principles of inheritance and variation, Mendelian genetics and the disorders by the Mendelian and inherited disorder or genetic disorder. Now today we are going to start a new chapter number 6, the molecular basis of inheritance. मीन्स जो हमने सीखा है कि अगले यूनिट में हमने सीखा है जो कि कैसे ट्रेट्स और कैरेक्टर फ्रॉम पेरेंट्स टू ऑफस्प्रिंग में ट्रांसफ़र होता है, उसका हम मोलिकुलर बेसिस देखेंगे. we will see at the molecular basis that what is the factor and what is the that molecules which transfer from parents to the offspring. So the chapter number 6, molecular basis of inheritance, which means what is the base of this inheritance or what are the molecules that are responsible for the inheritance. As Mendel said that there is one factor which is transfer from parents to the offspring. Later on many discoveries lead to the conclusion that the nucleic acid is the genetic material in all the living organisms as well as in plants and in animals. Then later in 1953, James Watson and Francis Crick discovered the structure of DNA, and this discovery leads to the tremendous booster in a branch of molecular biology and genetics. So here we will start first chapter number 6, molecular basis of inheritance. So what are the molecules which is responsible for the inheritance? After many experiments carried out on different molecules of the cell, we come to at the conclusion that the nucleic acid is the genetic material in all the organism, and this nucleic acid are DNA and RNA. What is DNA? DNA is deoxynucleic acid, where RNA is ribose nucleic acid. The DNA is found in most of the organism as a genetic material, where RNA is found in certain bacteria and prokaryotes. Another role of RNA is as a messenger and adapter, where DNA function as a genetic material only. Now let us see the structure of DNA. Today here we will discuss about structure of DNA, then the processes of DNA, that by which process DNA makes their copies and how they express in the cell. So this is known as a central dogma of molecular biology, which means they says that how this DNA expresses in a cell in a particular manner. Then we will discuss about different processes like replication, transcription, translation, and then we will also see that by which experiment it is concluded that nucleic acid is a genetic material. So let us start with the DNA, the structure of DNA. As we know that DNA is a deoxyribose nucleic acid. That can be asked in MCQ that what is the full form of DNA? DNA is a deoxyribose nucleic acid and RNA is a ribose nucleic acid. There are the many cells have different length of DNA. The length of DNA is vary from the cell to cell, from the organism to organism. For example, in bacteriophage, the length of DNA is 5,360 base pair. The length of DNA is expressed in a number of base pairs. So how many base pairs are present in a bacteriophage? In bacteriophage there is 5,386 base pairs are present. Where on the other hand, in haploid content of human DNA, the base pair are 3.3 multiplied by 10 raised to power 9 base pair are present in the haploid content of the human DNA. Now structure of DNA. DNA is composed of three components. First one is a nitrogen base. The another one is a pentose sugar, and third one is a phosphate group, which means DNA is composed of three components. First one is nitrogen base, another one is sugar, and third one is a phosphate. So the nitrogen base which is present in a DNA having two types. First one is a purine, and another type of nitrogen base is pyrimidine. So what is purine and pyrimidine? Pyrimidine is a nitrogenous base having two aromatic rings, where pyrimidine is a nitrogen base having single aromatic ring. The examples of purine is adenine and guanine. On other hand, the examples of pyrimidine is cytosine, uracil, and thymine. In DNA, adenine base with the thymine and guanine base with the cytosine. And in RNA, this adenine pair with the uracil. And in DNA, this adenine is paired with the thymine. Okay, so what is the difference between RNA and DNA? The basic difference in RNA and DNA is the RNA having single standard and DNA is a double standard. In RNA, the base pairs are adenine pair with the uracil, and in DNA, adenine make base pair with the thymine. Okay. Now another component in DNA is pentose sugar, which means sugar having 5 carbons. There is two types of sugar in nucleic acid. First one is a ribose sugar, other one is a deoxyribose sugar. You can see here, this is the pentose sugar having 5 carbon. Okay, first, second, third, 4 and 5. Pentose sugar having 5 carbons. The basic difference between ribose sugar and deoxyribose sugar is one oxygen is absent in deoxyribose sugar, where it is present in ribose sugar. Here in second carbon in RNA, here OH is present, where in DNA only H is present, O is absent. So now you can see that only just one oxygen makes how much difference in the genetic material and in the molecules. Here only one oxygen absent in deoxyribose sugar and present in RNA. So what is the role of this hydroxyl group, deoxyhydroxyl group? Here, due to this hydroxyl group, RNA become more reactive than DNA, and if this oxygen is absent it become deoxyribose sugar, and it is less reactive than RNA. So here DNA deoxyribose sugar gives stability to the DNA, because it have less reactive group at the carbon number 2, where in ribosome it have highly reactive group called OH. That's why they are not very stable, and deoxyribose sugar gives stability to the DNA or particular organism. It leads to the formation of heredity and passing of particular characters. So the evolution is the variation occur in DNA is quite less chances, and in variation occur in ribosome it is very high chances, and it is not stable because this hydroxyl group, highly reactive hydroxyl group. Now another component of nucleic acid is phosphate sugar. The third component is phosphate group, which is found on the pentose sugar. It binds with the pentose sugar and from nucleotide. Let us see what is nucleotide. So at the first carbon, remember that at first carbon nitrogen base will have, for example, adenine, guanine, cytosine and thymine, and at the carbon number 5 phosphate group will be present. Here phosphate group. This is known as nucleotide. Nucleotide having 3 components. First one is a nitrogen base, sugar and phosphate. If only nitrogen base and sugar is present, it is known as nucleoside. But when phosphate is added in nucleoside, here phosphate, it's called nucleotide. So what is the basic difference between nucleotide and nucleoside? Nucleoside is in nucleoside phosphate, only phosphate and nitrogen base is present, where in nucleotide phosphate group is added with the nucleoside. So this is about components of nucleic acid. Now let us see the actual structure of DNA, which is proposed by James Watson and Francis Crick in 1953 by the process called X-ray diffraction. And they proposed that DNA is made up of 2 chains which is anti-parallel to each other, where first chain go to the 5' to 3' and another chain go from 3' to 5'. So let us see the actual structure, ultrastructure of this DNA. Let us see this one strand only first. Okay. So if I zoom this for single strand, it will look like this, where we have seen the structure of nucleotide, where in nucleotide nitrogen base is present, is linked with the pentose sugar by the linkage called glycoside linkage, and on the carbon number 1, then carbon number 2, 3, 4 and 5. On the carbon number 5, the phosphate group is present. Okay, and wherever this phosphate group present in a chain, this end is known as 5' end. Where on the carbon number 3, having OH will bind with the another phosphate group and then it bind with the sugar. So it will make backbone or chain having phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. One strand having if strand having a phosphate group, it is known as 5' end, and having OH group known as 3' end. Okay, the two nucleotides is linked by the 3', 5' phosphodiester bond. Okay, so this bond is known as phosphodiester bond, which is two nucleotide bind with the phosphodiester bond, and this base pair bind with this pentose sugar by the glycosidic linkage. That can be asked in exam, that by which linkage nitrogen base pair with the pentose sugar. So the N-glycosidic linkage between present between the base and pentose sugar. Now, what is the silent features of this DNA?